Today we are going to study the enzyme structure, then cofactor, and then the classification of enzyme. First of all, we are going to look the structure introduction. Enzyme is a biocatalyst which speed up the chemical reaction. Enzyme is a three-dimensional globular protein, tertiary or tetrahedral protein. Some special species of RNA act as enzyme called ribose enzyme, or also called hammerhead enzyme. For example, hammerhead enzyme. It's highly specific in function. Structure contains structure called active site, which help in substrate and coefficient and prostatic binding. Also contain a residue which help in the substrate binding. Active site of enzyme made up 5% of enzyme. It has specific shape and structure, and it's specific in its function. Enzyme, a protein can cause a change in enzymatic protein can cause a change in structure and function. Active site of enzyme is divided into two sites: binding site, catalytic site. The binding site of enzyme is which select and bind with the substrate, and the catalytic site of enzyme is which catalyze the substrate bind with enzyme cofactor. Cofactor, the non-protein part of enzyme, which the non-protein part of enzyme, the non-protein part of the enzyme, which perform the function which cannot be performed by 20 amino acids. There are two type of cofactor: organic cofactor and inorganic cofactor. Organic cofactors are the organic molecules which are essential for the proper functioning of enzyme is called organic cofactor. Organic cofactor, like we have glycophosphorylase, which also which need pyruvate phosphate for proper functioning. Then we have inorganic factor. The inorganic factor is a molecule which is required for the proper functioning of enzyme. For example, for aldehydes enzyme need Zn++ and exokinesis need mmg++ for proper functioning. Then we have organic compound is further divided. Organic cofactor further divided into prostate group and coenzyme. Prostate group, the when in which the organic molecules are tightly bound, is called prostate group. For example, heme group and biotin. And coenzyme, in which the organic molecules are loosely bound. The molecule are loosely bound. It's called coenzyme. For example, NAD+. Then characteristic, they are specific in function. They are specific in function, structure, and sensitive to pH and temperature. If the pH and temperature is little bit changed, it can slow down or stop the enzyme functioning or denature them. They speed up the reaction. They add in the chemical reaction and did not change the end product but speed it up. They did not change the reaction. There are no harmful. They don't harm to the substrate. And the turnover the turnover number is the amount of substrate formation in the amount of substrate enzyme can form in 1 minute is called turnover number. Then we have formation. Enzymes are form when DNA transcript is translated by ribosome of rough endoplasmic reticulum, and then it's making. There are two type of enzyme: intercellular, extracellular enzyme. The intercellular enzyme are those which are made in cell and stays in cell for functioning, like we have oxidoreductase enzyme. Extracellular enzyme, the enzyme which are made in cell but they are transferred to other part of cell for proper functioning, like we have chylotrypsin, which made in pancreas but go to the intestine for proper functioning. Then classification of enzyme. The enzymes are classified by the international committee of enzyme. Enzymes are divided into 6 categories. They are categorized on the basis of classify on the basis of their function, and these 6 types are divided into subtype, and subtypes are divided as into subtypes. First of all, oxidoreductase. Oxygen and reduction is its function, and it's lactate and dehydrase. Then we have transferase, which help in transferation. We have hexokinesis in that. Then we have hydrolysis enzymes, bond cleavage with the transfer of formational group of with the bond cleavage with the transfer of functional group of water. It we have in this lysosome when lyase non-function group change its femur. Then we have isomerism isomerase enzyme, in which we have isomerization, uh, triphosphate isomers. We have then we have a ligase enzyme. The substrate bond form use ATP. The substrate bond form use ATP. Hydrolysis like RNA polymers. Then when we have pH of enzyme is almost 7, and the mixture pH is almost 7 too, when then CO minus minus and CO minus and NH3 plus NH3 plus is normal. It's a normal form. When the pH is lower, and then the our CO gain H, which change COOH into COH, and COH and NH3 plus remain the same. But when the pH is high, so then our NH3 lose and N and become NH2, like here I show. Then some enzyme pepsin is present in stomach and work in 4 pH. It help in digestion of protein. Then we have chylotrypsin, which is present in which is make in pancreas, and then straight to the stomach, to the intestine, which also work in 7 to 8 pH and help in the digestion of protein. When we have pepin, it's a it's again from tropical plant, and work proper in 7 to 8 pH. Then we have lysine. It's a microbugs, work properly in 5 to 8 pH, help in the digestion of olive oil. Then we have alpha glucose. It's also a microbulb, work in the 5.2 6.6, and help in the digestion of maltose. Then we have beta amylose, which is gained from maltose, and work in 5.2 pH and help in the denaturing of starch. Then we have glucose amylose fructokinase, again from tomato, work in 4.5, and Saccharomers and pectolyze microbugs 9.0 to 9.2, work in pectoral acid, help in the digestion of pectoral acid. Then we have xanthan oxide, obtained from milk, work properly in 8.3, and help in the digestion of xanthan. When we have lipogenesis, obtained from soybean, work properly in 9.0, and lysogenesis type 1, obtained from soybean, work in 9.0, 9.9 pH. And we have lipogenesis type 2, which is also obtained by soybean and work properly in 6.5 pH, and it's help in the digestion of lincolnic acid. That was it for